Good afternoon, students. Today, let's move to one interesting topic of complete biology, that is the DNA replication. We have already came to know that DNA is a double helix and is having two polynucleotides coiled around each other. It gives a look like this. So, this is one strand. This is another strand. So this is DNA having two polynucleotides. And the process of replication is what? How the DNA produces a copy of itself. That is the multiplication or reproduction of DNA. Now, we already came to know that the DNA is having two polynucleotides are antiparallel. If it moves in this direction, then the another strand moves in the opposite direction. How that happens? Let's have a little bit look of that. This is a nitrogenous base. Okay. So this is oxygen 1, 2, 3, 4. Here is the fifth one. Okay. So here is the another one, and this is the another polynucleotide. It will be having the head like this. Okay, 0. This oxygen 1, 2, 3, 4, 5. So what happens exactly here is in the fifth direction is 3PO4 get binded. Understood? H3PO4 get binded there, and here in the 3 direction. So here the orientation is based on 5' to 3' direction. So here 3' to 5' direction. That is based on the process we are finding it has 3' to 5' and 5' to another one is 3'. When I speak about the replication of DNA, we'll be knowing some of the most important aspect of DNA replication is DNA is a double-stranded structure. First thing is that is the nick. Number one, nick. Number one, you keep in mind nick. What is the meaning of nick? When you go to a tailor, when they make a shirt from the piece of cloth that you have given, they make a cut. That is called a nick. That is the point where the replication starts. Okay. Now, one enzyme called helicase. Helicase, number 2, helicase, which helps in breaking the hydrogen bond. So, this is the DNA strand. DNA strand, and along with the DNA strand, you see, there is the another DNA strand. So what this helicase does, helicase will be breaking the hydrogen bond. Here the helicase works and keep on breaking the hydrogen bond. So these two get separated and it is called replicating fork. Replicating fork. Understood the thing? Why it is replicating? Because it is giving, this is the strand of DNA. This is another strand of DNA, and it has given a fork-like structure, Y-shaped structure. Understood? This is the structure we are finding. So it is called a replicating fork, F-O-R-K, fork. Understood? So here I am writing 3' to 5' and 5' to 3'. This is the end where the replication takes. So for the time being, I am taking out this one. This is the 2 strand which has been separated and it has formed what? Fork. Understood? Next, we need to know another most important enzyme, topoisomerase. Third, topoisomerase. The function of topoisomerase is that it does not let the helicase to make too much of stress over that of the DNA. So it neutralizes the tension if it is exerted by the helicase, so that the DNA, otherwise it will be completely breaking. Understood the thing? Otherwise, what will happen? The DNA will be completely breaking into pieces, torn into pieces. And that topoisomerase helps in making it attention free. So, let's go for the another part now. This one done. Now, suppose I am going for the replicating fork. This is the one DNA and it has formed a fork here for replicating. This is fork, and these are the complementary nucleotide that we have done. Okay, these are complementary nucleotide. Here it will be acting. Now, first thing that comes, these two strands are not separated. Here remains, you see this part. Here I am putting some names. Okay, this is called SSB. What it is called? SSB. So it is called single stranded binding protein. What is called? Single stranded binding protein. This single stranded binding protein does not let the two separated polynucleotide chain or the two strands of the polynucleotide get united. It keeps separated. Okay. Next, another most important thing comes after that is the RNA primer. RNA primer. RNA primer is a small segment of RNA which is produced by permease. Try to get the word. Permease is an enzyme which helps in producing the RNA primer. So what happens now exactly? Here, suppose I am taking it to be 3' to 5', and here 5' to what? 3'. So RNA primer first of all will be coming and sitting here. RNA primer will be coming and sitting here. Now, in that case, another most important significant role, RNA primer, how it is produced? It is produced from the RNA with the help of permease enzyme. Then we are finding another most important enzyme called DNA polymerase. What is the function of DNA polymerase? DNA polymerase try to bring the nucleotide. Suppose here, when we are speaking about this DNA, this is the mother strand, is having the nucleotide, isn't it? This is having nucleotide. Now, when I have drawn this one, RNA primer has come. This is the RNA primer. This RNA primer also need to have nucleotide here. This nucleotide need to be complementary with that of the mother strand. This is called template. Remember another word, template. T-E-M-P-L-A-T-E, template or mother strand. So already the mother strand has the nucleotide, incoming nucleotide, who need to be complementary, sitting on the primer, will be decided by the DNA polymerase, who will be sitting in this point and will be selecting the complementary nucleotide. Understood? So this is done. Now see another thing. Two strands will be formed because the two strands here of the original DNA are separated. Definitely two new will be coming. One strand will be leading This is called leading 3' to 5', and its work is from 5' to 3'. It works from 5' to 3'. Another part, that is 5' to 3', this part, there will be fragments of RNA primer coming. Not a complete single strand as we found in this case. So definitely there will be complementary nucleotide coming based on the mother strand DNA template. Okay, complementary will be coming, which will be regulated again by whom? DNA polymerase. Up to here is okay. But it is a fragment. This fragment was seen first of all by a Japanese scientist who was called, or whose name was Okazaki, Okazaki. Okazaki. So it is called Okazaki fragments. Okazaki fragments. Okay. Now what happened? Another enzyme. I am telling another enzyme now. DNA Polymerase. DNA polymerase, what it will do? It will bind the fragments of RNA primer and make it one. Now this is also leading and this is also continuous. Leading was originally continuous, but it was a discontinuous, which was made continuous by whom? DNA ligase. Now what happens? Ultimately there will be calling. Here you are finding this is the daughter. This is called daughter strand. These two are the daughter strand, and this one is the mother strand. So ultimately what you are finding? You are finding this is the strand, and this the red one I am drawn here. This is the daughter strand. You are finding coiled. So it is a semi-conservative. DNA replication is semi-conservative. Ultimately what you are finding? I am rubbing this one. You see this one? I am rubbing this one. Complementary got done. So what you have found? You have found like this structure has happened. Ultimately this is I am rubbing again here. You see? What you are finding? DNA strand, double strand has been formed. Ultimately two strand has been formed. This is how the replication of DNA takes place. Okay? And it was the semi-conservative model that we have discussed, and it is the only proof that the semi-conservative process only works in the DNA replication. Thank you.